Good morning, everyone. Today we are going to talk about Pondguard. Pondguard is an essential oil blend supplement to reduce the disease risk and increase the productivity of aquatic animal, mainly the shrimp. Introduction, field observation, Indonesia field reports, field observation, overseas, lab reports, concept proving. First we come to introduction. This is how the packaging of Pondguard looks like. Pondguard acts as an immunomodulator. It maintains the general health of shrimp and helps in overall improvement in shrimp quality and productivity. It works directly against specific viral pathogens like WSSV and IMNV of shrimp. It works directly against specific bacterial and pathogens like Vibrio harveyi, Vibrio parahaemolyticus, and also the strain of AHPND of Vibrio parahaemolyticus. Composition, the selection of oil blend done based on antiviral properties, immunomodulating properties, and their combabilities to each other. The concentrate oil consists of three plant oil is a Eucalyptus, globulus, Pinus, sylvestris, Lavandula, latifolia. This concentrate oil is mixed 3 liter in 9997 liter of purified water to make 1 ton of the product. Synergism of essential oil blend concept. Synergism of blend. Many extracts were found to have a specific activity or activities by themselves. A blend of other extracts may enhance the required outcome. Bioactivity synergies between different preparation or compounds may improve the effect of a blend or extract. Synergism can be achieved through different pathways. Each essential oil have multiple terpenes and sesquiterpenes that can be biologically active. So at time, there will be multiple chemical reactions and multiple biological compounds working together to metabolize these compounds, produces multiple biologically active components to fight against pathogens, which act as immunostimulant or immunomodulator. It is inhibiting and interfering with the specific pathogen structures and proteins, such as membrane permeability. It enhances absorption of polar compounds across biomembranes, that is, carrier of active herbal blend to its target. It reduces toxicity of active compounds to the organism while keeping its activity against the pathogen. This can be seen with the graphics that how it works synergistically and have effects and each and every activities of the tissue. It works as anti-stress response, anti-free radicals, anti-inflammatory, antiviral, antibacterial green, colored, shown in that, and cellular signaling and apoptosis. Then how to prove this concept? So we use in silico or docking model. What are they? The natural antimicrobials could be effective in reducing viral and bacterial pathogenicity via direct or indirect antivirulence activities. The molecular docking or in silico model is a widely used technique to study a small molecular protein natural component in gradient interaction with the viral protein. So it's a protein-to-protein interaction at the atomic level. After calculating the binding position between the ligand and the target, the main purpose is to study the plant protein target viral protein interaction at the atomic level. This way, molecular docking methods allow a screening of entire libraries, allows a precise antiviral or antibacterial component or drug development and lead optimization. The in silico method also allows us to predict or help us to estimate the level of protection of each protein molecule present in the plant against the viral protein. In the present study, small molecule proteins of 3 plant oils, eucalyptus oil, pine oil, and lavender oils were studied against the targeted proteins of AHPND-EMS. The small protein molecules were downloaded from the PDB website and opened in the PyMol software. As a second step, the potential proteins of interest from the AHPND virus bacteria were downloaded and copied to the PyMol software. By scrolling and processing, it was allowed to bind both the molecules. To have a clear and fair study, the extra structures and water molecules of the protein were deleted by using the command. Only the interested domain of protein were used. The autoinducer molecules were removed and the molecule of interest were logged in. The protein structure was saved as LASR protein, which served as the receptor. So these are the steps: the multi-sequence alignment and the protein sequence similarity between Vibrio parahaemolyticus, PID-A, PID-B proteins, and the bacterial toxins. Protein homology modeling and protein-protein docking. The structural analysis of the cytotoxic mechanism of Vibrio parahaemolyticus, PID-A toxin. The structure-based ligand binding site and druggability prediction for Vibrio parahaemolyticus, PID-A, PID-B, and pathogens CRY toxins. Identification of Vibrio parahaemolyticus, PID-A, PID-B, and pathogens CRY toxin potential inhibitors in by structure-based virtual screening and molecular docking. So how it works, and then natural compounds present in Pondguard, including anethol, anisile, acetone, borneol, phenyl, ethyl alcohol, polypropylene glycol, and tripropylene glycol could be potent suppressors of four major potential pathogenic mechanisms of Vibrio parahaemolyticus induced AHPND in Pinus venomous shrimp, including PID-A, VP toxin interaction with Pinus venomous shrimp, APN, and ALP receptors, PID-A toxin homodimer, and PID-A, PID-B heterotetramer formation. Importance of essential oil. Yeah, as you see from the right side, it's in terms of scientific paper published yearly basis is keep on increasing. So there is much interest, and researchers all over the world are getting involved to find the utility and the work function, mechanism of action of the essential oil and herbal products. Indonesia is one of the topmost countries using herbs in aquaculture. Only in West Java, almost 46% of the farmers use herbs in their ponds. In India, several companies produce specific herbal extracts based remedies for aquaculture and livestock, mainly as feed additives. In China, antibiotics in aquaculture have been modified by adding herbal extracts to the feed or directly to the water to improve fish or shrimp's immune system and digestive system. The figure also demonstrates on the right side the popularity of herbs and essential oil as an alternative and substitute of drugs in aquaculture and animal husbandry. We have two kinds of products. One is concentrate, which we call Pondguard oil, and the diluted ready-to-use is Pondguard. This is the difference between these two, and depends on the convenience it is supplied. This is a dose of the Pondguard in application in water. So this is before post-larvae stocking in the pond, -7 days before stocking, -3 days before stocking, then during culture, at day of culture 1 until harvest. So we use 0.4 ppm. Frequency is once a week, and during culture, it goes 2 times a week. If there is emergency case like a stress condition, DO drop, plankton crash, or appearance of disease in the nearby area or in the pond, then we have to apply emergency dose, which is 2 times a day for 5 to 7 days or until mortality slows down. Then we come to the shrimp performance in Indonesia, in particular. what we see that this is before application of Pondguard and included in our SOP. After adding in the SOP, we can see that we can have, lower FCR, better survival rate, productivity is now 20 ton per hectare compared to 19-18 ton. And this is, also showing that the mean body weight is increasing. Then we talk about some disease incidents, and we see that like IMNV, WSSV, AHP, and whitefaces, all the incidence rate are reducing in the ponds and in the culture system. And if we talk about the overseas, so this is first where we see that very clear we can increase the or keep the survived shrimp in the ponds and so have better harvest. This was in WSSV infected pond. This is a big area, 10 hectare pond area, where again we can increase the productivity, and the final performance index was better, 1.68 versus 1.54. This is the third trial where we got the better FCR, tonnage, and survival rate almost 6% different. Now this is in another country where we see that Pondguard application, where our main target was to reduce the number of WSSV cases and IMNV and enhance the productivity. So here we see that we could perform better in FCR, 1.1 versus 1.32, and the survival rate, 85% versus 65 in the control. The second on monodon, what we see that we have better FCR, 1.2 versus 1.3, and very good survival rate, 87% versus 65. And along with this, it can also protect from WSSV in those areas. This is the third one in the southern part of India, where we could get almost, 90% survival rate compared to 76. And so the biomass was 2.5 ton versus 1.9. FCR was lower too. This is in China, where our trial was conducted in EMS AHPND prone area, and we could make pond survive or delayed harvest. And then we have some lab trial reports, which is on to prove it's an immunomodulator, then anti-WSSV, anti-IMNV, anti-AHPND, anti-Vibrio, and then overall growth performance, and then its impact on live food. So this is showing by the hemocyte count that it works as an immunomodulator. Then this is against in vivo trial was conducted in the lab to show that it works against WSSV infection. The third one was conducted in Vietnam, and the trial showed that Pondguard application was able to prevent the horizontal transmission and also to protect shrimp from mortality against AHPND. This is infectious myonecrosis virus, where we could save up to 100% of the shrimp. This is on the green Vibrio, which we see that after 24 hours, the Vibrio count goes down, whereas in control, it still persists. Then we talk about how is the impact of Pondguard, because when we are showing you that it works against virus, bacteria, then the question comes whether it also kills the helpful bacteria or the live, live food present in the water. So we conducted several trials, and this is our common probiotics bacteria, Bacillus, Lactobacillus, and Rhodobacter. So there is no negative impact. No inhibition zone was, formed. The second, again, it is showing against Bacillus species. that it has no negative impact on the growth of bacillus. But we also tried on pathogenic Vibrio, which showed that it is having the inhibition zone. So the other side, if it is Vibrio harveyi species, which are highly pathogenic, it inhibits the growth of this bacteria, luminescent, also the Vibrio harveyi and photobacterium. Then on the live food, we tried with three common items: Thalassiosira, Ketoceros, and Tetraselmis, with the higher doses, 0.6 ppm and 0.8 ppm. And we saw that there is no negative impact, and all the groups grow same as control. So in conclusion, the developed blend formulation, Pondguard, is an effective immunomodulator and anti-pathogenic agent which supports shrimps to grow in its optimum condition, which resulted in better productivity. It has no negative effect or less negative effect on the live food and probiotics bacteria. We have some testimonies and publications. This is from India on Vanami. We have some testimonials showing that it has protected against IMNV and have better survival and productivity. This is Monodon, having better productivity. This is in the southern part, Andhra Pradesh. These are the testimonies where we have better survival and productivity. This is in Indonesia. It's in Saudi Arabia. It's in Vietnam. It's in China, where mostly, indoor systems are working. These are some of our publications. Then I would like to special thanks to CEPHAS, Vietnam, Can Tho University, Vietnam National University of Agriculture, IPP, Bogor, Shanghai Ocean University, China, and NAQUA, Saudi Arabia, Mayang Kakwa, India, Pangfei, China, and our CPP management and rear management. Thank you very much.